everyone. Today we will see some questions from cell cycle and cell division. Hope you like it. So the first question is, in cell cycle different phases are in the following sequence. So choose the correct sequence. We know that in cell cycle we have mainly 4 stages, that is the G1 stage, S stage, G2 stage, and then the M stage. So this is the sequence. We have this G1 is called as gap 1 phase, and then S is the synthesis phase, then G2 is the gap 2 phase, and then M is the mitotic phase. So this will be the sequence of it. So here if you see here, the option B is the correct answer. G1 phase, S phase, G2 phase, and the M phase. So second question is, in which phase proteins for spindle formation are synthesized? It's not in the G0 phase, not in the G1 phase, not in the S phase, but it's in the G2 phase. Basically G2 phase is where the spindle fiber formation proteins occur. So G2 phase is also called as post synthetic phase. Why we are calling it as post synthetic phase? If you remember, G1, S, G2, and then M phase, right? So it is coming after the S phase. So that's why we are calling it as post synthetic phase, or we also call it as pre-mitotic phase, because it comes before the mitosis. So G2 phase, the other name of G2 phase is the post synthetic phase, and then the pre-mitotic phase. Third question, which of the following can be classified as G0 cells? So we know the cycle very well: G1 phase, S phase, G2 phase, then the M phase, right? So few cells, after they are differentiated, they undergo G0 phase. So after the mitotic division, instead of entering into the G1 phase, they enter into the G0 phase. So here let's see something about the G0 phase. So G0 phase is basically the resting period of the cell, or we also call it as quiescent phase. That means both, it's the same. Resting period or quiescent is the same. The thing you have to remember about the G0 phase is, here the cells are basically active. They are metabolically active only, but it's just they are not dividing, that's it. So division is not occurring. So that is the thing. Just because they are not dividing does not mean they are not active. They are metabolically active, but the division does not occur. So the examples are given here. So neurons, fibroblasts, lymphocytes, and then the monocytes. So in all these 4, the correct answer, the cells which are undergoing the G0 phase is the neurons. So all these 3, they divide, they continue division, but whereas the neurons, they undergo the G0 stage. Fourth question, the time period between meiotic 1 and meiotic 2 cell division is called growth phase, first gap period, interkinesis, or interphase. So the correct answer is the interkinesis. Fifth question, the drug colchicine has inhibitory effect on cell cycle in which stage? So what do you mean by inhibitory effect? That means which is interfering or which is stopping. So let's see what does the drug do on which phase, and then you can answer it on your own. So the drug colchicine it basically interferes with the formation of microtubules. Where do we require these microtubules? In which phase? That is in the M phase or the mitotic phase. For example, if you take this is the cell, okay, and which stage am I drawing? Anyone can answer? That is the metaphase. Specifically speaking, it will be the metaphase, right? So here we have, these are the centrioles on the opposite sides. From that we have the spindle fibers coming and attaching to the centromere. Now the drug colchicine interferes with these spindle formation, spindle fibers, because these spindle fibers are formed from the microtubules, right? Now because these microtubules are interfered, this spindle formation does not occur and it is arrested in the metaphase. Okay, specifically which phase in M phase means it will be the metaphase. Okay, so let's go back to the question. The drug colchicine has inhibitory effect on cell cycle in which stage? So now you can answer, it will be in the M phase. Specifically speaking, it will be the metaphase, and for this very reason it is also called as mitotic poison. Okay, so this drug colchicine is called as mitotic poison. Sixth question, which of the following does not occur in the interphase of eukaryotic cell division? So increase in ATP synthesis occurs, increase in DNA synthesis occurs, increase in RNA synthesis also occurs. Reduction in cell size does not occur. Basically the size of the cell increases, it does not reduces. So the correct option will be the option D. Seventh question, how many generations of mitosis are required to produce 128 cells from a single cell of root tip? So we have to see the mitosis here. So in mitosis we know one single cell gives rise to two cells. So here again each cell will give rise to two cells. So this keeps on continuing, right? So here they're asking one cell, in the end it gave 128 cells. So how many generations did it take? So here we can use the formula 2 power n. 2 power n, that is 2 power n is equal to 128. So we need to know what number we should add in the n so that it will be equal to 128. So that will be 2 power 7 is equal to 128. You can just check it. 2 power 7 will be equal to 128. So the correct option for this is option number C. Eighth question, in mitosis, nucleolus and nuclear membrane disappears at which phase? Metaphase, interphase, telophase and prophase. So the correct answer is prophase. So in the prophase only the nucleolus decreases in size and gets disappeared, and by the end of the prophase what happens is the nuclear membrane also disappears. Eighth question, in mitosis, nucleolus and nuclear membrane disappears at which phase? Metaphase, interphase, telophase and prophase. So the correct answer is prophase. So in the prophase only the nucleolus decreases in size and gets disappeared, and by the end of the prophase what happens is the nuclear membrane also disappears. Ninth question, early prophase is also called spireme stage. In this stage, chromatin fibers are undergoing spiralization, chromatids are twisted over each other, chromosomes are spirally twisted, chromosomes are overlapping one another. The correct answer is chromosomes are overlapping one another. To talk about the spireme stage, so spireme stage is a distinct characteristic of early prophase. In early prophase we can see it. So this is the cell, then this is the nuclear membrane, right? And then we have the centrioles which are getting divided and moving. right? So inside this nuclear membrane, the chromosomes are overlapping. So the chromosomes are overlapped upon one another. So you cannot, so you basically cannot, they're not distinct. They're not at all distinct. That means you cannot tell that this chromosome, okay, and it has this ball of wool appearance. So in which stage you see ball of wool appearance means it is in the spireme stage. Because of this it is called as a spireme stage, which is in the very early prophase. So let's go back to the question. So that's why the chromosomes are overlapping one another. 10th question. Tetrad is made up of 4 homologous chromosomes with 4 chromatids, 2 homologous chromosomes each with 2 chromatids, 4 non-homologous chromatids, 4 non-homologous chromosomes. So let's see what is this tetrad. So let me draw the chromosome. We know chromosome is X shape, right? And then we have the another chromosome here. So let's say this is chromosome number 1. So we get one from the father and the other from the mother, right? Now this X shape is what we call chromosome. And then what is chromatid? Chromatid is, if you take the half of this chromosome, this one is a half of it, or a shape like this, okay? So this will be your chromatid. Now these two are chromosome 1, so that's why we call them as homologous, right? Why we are calling it as tetrad? Tetrad means what? Basically 4, right? So here you can see that here we have 1 chromatid, 2, 3, and then the 4. So that's why we call it as tetrad. We have 4 chromatids, right? Chromatid. So that's why we call it as a tetrad. Or there is one more name too. Let me erase this. So or else we also call it as a dyad. It's also called as dyads. Dyads means 2, 2. Okay. So let's go back to the question. So tetrad, now tetrad is homologous, 2 homologous chromosomes, right? 2 homologous chromosomes each with 2 chromatids. We don't have 4 homologous chromosomes, so it's wrong. Not non-homologous, so completely wrong. So the correct answer will be this option number B, 2 homologous chromosomes each with 2 chromatids. Let me clear this. So B will be the correct answer. 11th question. Read the given statements A and B and select the correct option. Statement A, synthesis of DNA takes place in S phase. That's true. Statement B, every chromosome during metaphase has 1 chromatid. We know that chromosome means it is always having 2 chromatids, not 1 chromatid. So this is statement B is wrong. So the correct answer for this will be statement A is correct and B is incorrect. So the option B. 12th question. Mitotic anaphase differs from metaphase in possessing. Okay, let's see this metaphase and anaphase so that you will be able to answer that question. So let me draw a cell here and the chromosome which is in the equatorial plate and another chromosome. Okay, then we have centrioles and then the spindle fibers attaching to the centromere, right? Now as you can see here, we have, this is the chromosome 1 and then this is the chromosome 2, right? We can tell that in this metaphase metaphase, it has 2 chromosomes, right? And then what else it has? 4, sorry, and it has 4 chromatids, correct? It has 4 chromatids. Now let's move to the anaphase. Okay, this is your anaphase, right? Now here what happens is the chromosomes are dividing, right? The chromosomes are dividing in the anaphase, that is this centromere is divided. So the centromere is divided here and they're close to the centrioles. Now you can see this is chromosome 1 and then this is chromosome 2, right? Now you can tell here in anaphase also it has 2 chromosomes, but how many chromatids did it have? It has only 2 chromatids, right? That means in both anaphase and metaphase chromosome number is same, but the chromatids became half. Okay, remember that the chromosome number is same but the chromatids became half. Let's go back to the question. So mitotic anaphase differs from metaphase in possessing same number of chromosomes and same number of chromatids, half number of chromosomes and half number of chromatids, half number of chromosomes and same number of chromatids, same number of chromosomes and half number of chromatids. What will be the correct option? That is option number D. It has the same number of chromosomes and half number of chromatids. 13th question. During cell division in apical meristem, the nuclear membrane appears in. This is very easy. So nuclear membrane will appear in the last stage, that is your telophase, right? In M phase we have many other, like we have 4 more, right? We have prophase, then metaphase, then anaphase, and then the telophase. So this is the last phase. So in the telophase you will get the nuclear membrane appearance. Next question. Spindle fiber unite with which structure of chromosome? This is very easy. Chromosometer? No. Chromomere? No. Centromere? Centromere is the correct answer. We know that we have chromosome here. In the chromosome in the center we have a structure called centromere, where the spindle fibers from the centriole come and join in the either side, right? So this structure is what we call it as centromere. 15th question. Best material for the study of mitosis in laboratory is? Very easy. It's the root tip, right? Root tip. Why we are choosing root tip? Because in root tip there is a rapid division, cell division, right? Because of this rapid cell division you can very easily study the mitosis. So that's why we go for root tip. Basically we use this onion root tip very commonly, very common and easy to grow. So we use the onion root tip. 16th question. Cell plate formation starts from phragmoplast, epiplast, tonoplast, protoplast. The correct answer is phragmoplast. So basically phragmoplast is the it gives this structural framework for the cell plate. It gives a structural framework for cell plate. So this is, by the moment you see the cell plate you need to understand that it is very specific. It is very specific in the plants, right? So you find this phragmoplast in the plants. 17th question. The major event that occurs during the anaphase of mitosis which brings about the equal distribution of chromosomes is? So in the previous question also we have seen that in metaphase we have chromatids joined together, but whereas in anaphase They are separated. Why they are separated? It's because of the splitting of the centromere. So the correct answer is splitting of centromere. 18th question. If you are provided with root tips of onion in your class and are asked to count the chromosomes, which of the following stages can you most conveniently look into? So it is metaphase, because in the metaphase all the chromosomes are arranged in the equatorial plate, so it is easy to count the chromosomes. So we basically choose the metaphase stage. 19th question. Who discovered mitosis in plant cell? Fleming, Van Beneden, Strasburger, Rudolph. The correct answer is Strasburger. So he is the person who discovered in the plant cell in year 1890, and mitosis in animal cell is discovered by Fleming. So he did it in animal cells in 1882. 20th question. There is no cell division of cytoplasm after mitosis. The condition will be, that means cytokinesis is not there. Only karyokinesis is there, right? So that means within the cell we will have two nucleus, right? So what will be the option? One cell with two nuclei. So that will be the correct answer. 21st question. Select the correct sequence of stages of meiosis to prophase. Either meiosis 1 or meiosis 2 prophase, it will be the same. Just have to know the stages. That is, I used to remember like LZPDD. That is leptotene, zygotene, facitine, diplotene and diakinesis. So the correct option of this will be option B. Right? Leptotene, zygotene, facitine, diplotene and diakinesis. 22nd question. Genetic recombination during meiosis takes place in which stage of prophase 1? Zygotene, diplotene, facitine, metaphase 1. So first we will talk little about this genetic recombination. So genetic recombination occurs between homologous chromosomes, right? So these are homologous chromosomes. Just to differentiate, I have drawn in two different colors. So what happens here is a small segment of this chromosome gets exchanged. Okay, so in this way what happens, the genes are getting recombined or getting combined. So that's why we call it as genetic recombination. Now this step happens exactly in the middle of the prophase 1. So if you remember L, Z, P, D, D, right? Exactly in the middle. That P means facitine. So let's see, the genetic recombination occurs in the facitine. That is option number C. Formation of bivalent during meiosis occur in. So let's see what is this bivalent. It is nothing but a pairing of the homologous chromosomes. That's it. So these two are homologous chromosomes, right? So now this pairing is what we call it as bivalent. This is bivalent stage, or also they call it as tetrad stage, right? Now this is this you can observe in the zygotene. That is the second stage of the prophase. Zygotene stage, second stage of prophase. 24th question. During diplotene stage in meiosis, chromosomes replicate, chiasmata shift towards the end of the chromosome, homologous chromosomes pair, chiasmata are seen. The correct answer is option number B. The chiasmata shift towards the end of the chromosome. 25th question. Which statement best explains the evolutionary advantage of meiosis. Meiosis are necessary for sexual reproduction. Genetic recombination are possible from generation to generation. Meiosis alternates with mitosis from generation to generation. The same genetic system is passed on from generation to generation. And the correct option will be the B, because of the genetic recombination only we have so many like new species, like the evolution is occurring because of this genetic recombination. So the correct option will be B. 26th question. When paternal and maternal chromosomes change their materials with each other in cell division, this event is called, told you before, it's called as genetic recombination, or we also call it as crossing over. So the correct answer is option number D. 27th question. Synaptonymal complex is found in Lambrusch chromosome, polytene chromosome, paired meiotic chromosome, mitotic chromosome. So the correct answer is paired meiotic chromosome, because this complex is required for recombination. You can see the recombination in the meiosis. So that is the pair. The option will be number C. So now let's look a little bit about the Lambrusch chromosome and polytene chromosome. Lambrusch chromosome, if you see this, you can find it in the vertebrate oocyte, but the exception is mammals. There is an exception. You don't find it in the mammals. And then why the name Lambrusch is because it has this brush like structure. You can see in the chromosome, because of this brush like structure only the name Lambrusch is given. Basically they are very much larger as well. What is this? They are very much larger as well. Large chromosomes. Okay, now this is about the little bit about the Lambrusch chromosome. And then coming to the polytene chromosome. Polytene chromosome I'm very well aware, because we used to observe it in the masters. So these are also called as giant chromosomes. Okay, so how do we get the polytene chromosome is, the polytene chromosome, if you see the structure, it is because of the repeated DNA replication. Okay, so what happens is, this is the chromomere. So the DNA gets replicated but it is not divided. So because of that reason you have so many bands. Okay, this is called as polytene chromosome. You find these in the insect larva. In the insect larva you can find this polytene chromosomes. These are also called as giant chromosomes. We used to observe it in the Drosophila larva during our master. So it's very interesting chromosome. 28th question. Read the given statements A and B and select the correct option. For a particular character in an individual, each gamete only gets one allele. That's correct. So statement A is correct. So only one allele enters into the, or it gets separated into the gametes. Then statement B, chromatids for a chromosome split and move towards opposite poles during anaphase of the mitosis. That is also correct, right? So during the anaphase only we see the splitting of the chromosome. So the correct option for this will be both statements A and B is correct. 29th question. Which of the following is the longest phase of the meiosis? This one everyone can answer by now, because that will be prophase 1, because in prophase itself we have 5 more sub stages, right? Leptotene, zygotene, facitine, diplotin, and diakinesis. So prophase is actually a complex phase and it is the longest phase in the meiosis. 30th question. Meiosis in capital A small a, capital B small b will result in gametes. So here we know this independent assortment, right? Very well. So they are independently assorted. So let's see, capital A small a, capital B small b. So they can have a combination of capital A small capital B, capital A and small b, small a and capital B, and small a and small b. So all these combinations of gametes can occur. So where is our answer in this? So it's in the option number C. 31st question. Condensins are majorly associated with anaphase, prophase, telophase, metaphase. The correct answer is prophases. The condensins are mainly used for the chromosomal condensation. So these are proteins. So condensins are proteins. So you can find these condensins everywhere from the bacteria to eukaryotes. And they use energy, that is the ATP, to condense this long chromatin, what we see, right? It's a long fiber like structure, chromatin, and convert it into a condensed chromosome. So that is the function of the condenser. 32nd question. G1 cyclin is destroyed at the end of which phase? G1, G2, S phase, or G0 phase? The correct answer is S phase. So this G1 cycle, if you see, this is required, it acts as a restriction point in the cell cycle. So after this S phase, by the end of this S phase, only when the G1 cyclin is destroyed, the cell will enter into the G2 phase, or else it will not enter into the G2 phase. So that's why G1 cyclin is destroyed by the end of the synthesis phase. 33rd question. Amitosis is a dash cell division process: direct, indirect, equational, and reduction. The correct answer is direct cell division process. Why we are calling it as direct? Because it's just a simple splitting of nucleus and the cytoplasm, simple splitting of it. And then here you can see there is unequal, random distribution of the genetic material. So unequal distribution of genetic material. And then you mostly see these amitosis in the unicellular organisms. For example, you can take some few bacteria, and then even in protozoans like paramecium, you can find this amitosis. 34th question. Cell plate grows from wall to center, center to wall, one wall to another, all of this. The cell plate always grows, remember, it's from the center to the wall. If this is the dividing cell, then the cell plate will start in the center and then grows towards the wall. And this type of cell plate we call it as, it's called as centrifugal cell plate, which goes from the center towards the wall. 35th question. Among 4 gametes formed in the end of sexual reproduction, the number of gametes not showing genetic variation due to crossing over is 4, 2, 3, 1. The correct answer will be 2, right? If you have a chromosome here, homologous chromosomes, right? Now only 2 of them are undergoing the genetic recombination, right? So now what will happen in the end? You get four cells. So let me draw 1, 2, 3, 4. So out of these only 2 are getting. I hope you're understanding. This is very difficult to draw. Yeah, so only you can see that only this one, and only this one are getting recombined, and these two are remaining the same, right? So the option will be 2. So 2 of them are not showing any genetic variation, only 2 are showing the genetic variation. 36th question. Reason of chromosomal movements in anaphase is astral rays, centrioles, kinetochore, spindle fibers. Please write your answers in the comments.